Hi everyone, my name is Haliza and I'm here to present my work on analysis of differentially expressed genes and molecular pathways in spontaneous preterm birth, a bioinformatics approach. So preterm birth is defined as birth before 37 gestational weeks and is one of the leading causes of infant mortality and morbidity. Thus far, approximately 35% of preterm infants die within 28 days of life. However, prevention efforts as of yet are very lacking due to incomplete understanding of the molecular mechanisms accountable to preterm birth, as the differential gene regulation might pinpoint the pathophysiological processes involved within a condition. Studies focusing on gene expression might enable us to understand the molecular mechanisms of preterm birth. As gene expression also varies across tissues during any disease formative stage, it is also crucial to identify gene regulation across different tissues to understand the etiology of preterm birth. So in this study, we aim to identify differentially expressed genes across maternal blood and placenta to understand the etiology and molecular mechanism involved in preterm birth. First, we conducted DEG analysis of chosen gene regulation studies according to the inclusion criteria stated here using Lima-R package. The DEGs are considered significant if they had p-value less than 0.05 or at least a two-fold change compared to term birth. The DEGs found from these individual studies are then integrated together according to the tissue utilized, either placenta or maternal blood, using p-value combination method to form meta-DEGs. Then we constructed a network based on these meta-DEGs and ranked them through their topological parameters using two cytoscape plugins, which are CytoHubber and CytoNCA. We compared the results of these two methods and obtained the commonly high-ranked genes. These genes were then compared with the ENCODE clusters to see which of them are located within a cluster, sharing similar biological pathway. The enriched pathway of these genes are then evaluated using enrich-R, and finally, the significance of these genes in preterm birth were validated using signature tool of Gene Investigator, which provides us a condition that has similar gene regulation signature as to the one obtained in this study. So here, figure 1 shows a pie chart of the total meta-DEGs obtained in this study, in which case we obtained 1,113 maternal blood meta-DEGs and 768 placenta meta-DEGs. Among them, a total of 29 maternal blood and 46 placenta meta-DEGs were commonly highly ranked by the two cytoscape plugins, where 20 of the maternal blood and 18 of the placenta meta-DEGs were involved in a cluster, being a part of similar biological pathways. Among these genes, the most centrally located genes are these for maternal blood and these for placenta, and we deem them as hub genes for this study. So the enrichment analysis of the genes involved in a similar cluster showed cytokine-related process for maternal blood, such as cytokine-mediated signaling pathway and cellular response to cytokine stimulus. Correspondingly, the KAGG pathways obtained are IL-17 signaling pathway, which itself is a pro-inflammatory cytokine, and T-cell receptor signaling pathway, which is involved in the synthesis of cytokines. On the other hand, placenta are more involved in extracellular matrix organization and pathways of ECM receptor interaction. Verification using the Gene Investigator tool provided us with the top 50 studies of similar expression patterns as to the 20 blood and 18 placenta gene signature obtained from the cluster analysis. For blood, the conditions shown are irritable bowel syndrome, IBS, Parkinson's disease, and ovary cancer, among others. For placenta, the conditions obtained are endometrial cancer, ovary cancer, and IBS, among others. So the results obtained in this study coincide with previous studies of preterm birth, especially the results of Gene Investigator, which provided conditions such as IBS, endometrial, and ovary cancer. These are one of the risk factors of preterm birth. And in overall, the maternal blood DEGs are involved in cytokine-related process where pro-inflammatory cytokines are triggered in response to bacterial toxins, which then synthesize secondary mediators such as prostaglandins promoting preterm delivery. And all three hub genes postulated in this study were involved in the synthesis of cytokines. And in placenta, the importance of ECM is highlighted. So ECM is responsible for maintaining the uterine structural integrity. And the overall ECM stability will be affected when there is even a slight change leading to adverse outcomes, including PPROM, cervical insufficiency, and uterine rupture. And similar to the maternal blood, the three hub genes postulated in this study were involved in the regulation of ECM. So each hub gene identified in this study may contribute to preterm birth in their own way. Hence, they should be considered as probable biomarkers and studied further. That's all from me. Thank you.